And finally, last but not least, Jason is going to talk to us about Enigma and what again the...
After the association study. So yes. After your association study.
After the association, all this interpretation aspect of the biology that we need to put in. Yeah.
Right.
Thanks, Jason.
Great. So last talk. So the idea here is that you performed a genome-wide association study, you performed a rare variant association study, you've identified some locus in the genome that you're interested in. And so a point that I want to make very strongly is that a hit is just the beginning. Even though we, for example, in Derek's study, we identified a particular genetic variant which is strongly associated to putamen volume, we have only identified variation in a locus of the genome which significantly influences our trait, in this case putamen volume. We have not identified a particular causal variant, we have not identified a particular gene, we have not identified a causal biological pathway or a causal brain region. Really, I mean, it could be affecting some other region first, primarily, and then secondarily affecting this region. So what we have found by doing this genetic association study is very important, right? This is a causal foothold into understanding what the biology of this trait is, and something that, by doing it in an unbiased way, shows something that is totally new to biology. However, what we want to know is all these other things. We want to know what these causal genes are. We want to know what the causal variants are. And most importantly, we want to know a mechanism by which that base pair change, or that particular type of genetic variant, influences brain structure, function, or risk for disease. Okay, so that's our goal. And I'm going to talk about some bioinformatic tools to help you understand better your traits. Although I'm going to talk about these bioinformatic tools, I think David's point is really important, that really you should do experimentation on the locus of interest to really prove the causality of that locus and to prove all of these different things, the causal variant, the causal gene, and causal biological pathways. Okay, so we've spent all this time finding a genetic association, and we want to interpret that genetic association. But first, before we go into any interpretation, we want to be very wary because it's extremely easy to make a story about any genetic association. I do it in the lab all the time, sometimes for fun, sometimes I think I really do understand it. So it's important to be aware of this. So just other authors have talked about this too. Human genomes have a high level of narrative potential to provide compelling but statistically poorly justified connections between mutations and phenotypes, right? This one I like a little bit better. A critical challenge for biologists will be avoiding premature hypotheses born of biological plausibility and just so stories. Some of my favorite stories, they're really good, but maybe don't make so much sense. And then findings from single association studies constitute tentative knowledge and must be interpreted with exceptional caution. This means you should replicate what your findings are and replicate them at each level of biology that you're studying. So importantly, biological plausibility is not a substitute for statistical significance. Follow the statistical significance. Biological plausibility is great to help you understand what's going on, but follow your statistics. That will help you really understand what's going on. Okay, so I'm going to talk about exploring biological mechanisms at different levels. I'm going to give you a lot of practical tools that I use consistently to understand or to try to get ideas about the genetic locus that we've been looking at. Hopefully they'll be useful to you as well. The first will be just exploring the genetic locus of interest. Next we'll talk a little bit about epigenetics. We'll try to move from the locus to gene, which is a problem that David just talked about. We're going to explore the expression of that gene in brain, and also look for enrichment in biological pathways. Okay, so genetic hits. So there's multiple types, as Sven and David talked to you about, multiple types of genetic mutations, and they can have different types of functions. So at the top there, this isn't working that well, but at the top here, you can see this is an exon, a coding region of the genome, and there can be mutations in this exon that can cause changes in the base, in a base pair that cause changes in the amino acid structure. They can cause inversions or deletions, which can alter the open reading frame and cause frame shift mutations. They can be nonsense mutations, which can cause an early stop to that protein. They can alter the splicing of that protein, or they can be in basically in areas that we can't really interpret very well yet, in intronic or intergenic regions. So these ones are really nice for interpretation, the ones in the exon, but the ones that are outside the exon are very hard to interpret. And unfortunately for us, for common variants, 88% of significant GWAS, or common variant loci, are often found in intergenic or intronic genes with no clear gene of action. So even though we identify this locus, it does not give us a gene of action. GWAS loci tag very large regions with multiple functional elements within them. So just to give an example, from the schizophrenia GWAS, the max locus size was 800 kilobases. For the mean size for that schizophrenia GWAS locus was 171 kilobases. And to give you a measurement of scale, the mean gene size from GenCode is 29 kilobases. So multiple genes can fit within one locus. So rare variant mutations, on the other hand, like David talked about, including missense or nonsense mutations, have a clear gene of action. So you can study them with better understanding of what the gene will be, but they are, as they say, rare. Okay, so I'm going to give you some practical usage to explore a genetic locus of interest. One of the most useful tools in all of genetics is the UCSC Genome Browser. It is your map. It will help you look at any particular region of the genome you're interested in, and will also help you zoom into particular features of the genome. So the UCSC Genome Browser is found at this website here, or just Google it. You can look at any genome of interest. You can look at anything from human to Ebola. You can select your group, your genome, and as Sarah talked about, the assembly is extremely important. So the assembly is the coordinate system that you use for looking up your genomic coordinates. The most commonly used as of today is HG19, even though the newest one is HG38, I think. They just skipped up to 38. So most people use this coordinate system, but it is not the newest and will not be the most used system in the future. So keep in mind which coordinate system you're in. You can then type in the search term either SNPs or genes or whatever kind of feature of the genome you're interested in. So we type in a particular hit. This is the top putamen hit, and I'm just going to go through multiple aspects of exploring the locus of this top putamen hit. So we type in the SNP of interest, and then we get a page resulting like this. And we get multiple things in this page. First is the ideogram, so it shows you in the chromosome and where you are in the chromosome. So there's a little red box there on the ideogram. That's where you are in that chromosome of chromosome 14. Next, you'll get a scale bar. So the scale bar here shows that you're at 200 bases from that. And importantly, again, shows your coordinate system. You're in HG19. After that, you can see the actual coordinates of particular base pairs. And then following that, you see different tracks. So all of these represent different aspects of the genome that you can annotate, right? And so most of these you can see are actually empty. So in RefSeq genes, there's actually no genes. For example, DNase clusters, there's no, I'll explain what those are in a minute, but there's none of those. But I can see at the very bottom there the actual SNP that I'm interested in. And so I typed in that SNP, and I'm looking at a very close region of the genome which contains that SNP. So what I want to do now is zoom out. Oh, I can also click to shrink that track. I want to do now is zoom out and look. Okay, so now I zoom out. The variant I initially entered stays in the center of the genome browser, and I can see now actual genes. So these are interesting functional elements. There are multiple isoforms of those genes that are contained there on the left-hand side. On the right, you can see other genes, and including one that's a long intergenic non-coding RNA on the right-hand side. And you can notice the scale at the top. Now I'm at 50 kilobases versus 200 bases that I was at before. Okay, so I have a variant in the center, but I'm interested in, if that variant affects any particular genes, and I haven't really demonstrated that yet, right? Okay, so another thing I may want to do is to add other tracks to explore what is the potential functionality of this variant by looking at, for example, what other GWAS have found. So there have been many GWAS done on many different types of traits, and I can add those tracks to the UCSC Genome Browser by just clicking below and clicking pack and then adding a track. And now I can see that in this I have a new track which shows genome-wide associations previously done. So interestingly, there's a hit that's found previously, so I can click on that hit to find out about this variant. to know what phenotypes were used to study it. So this is exploring that hit after I click that button. So I can see the SNP that was associated with that, and I can see the publication was about chemotherapeutic agents and severe neutropenia and leukopenia, which I know nothing about, but that could be potentially interesting. However, the initial sample size was 106 subjects, 106 cases, and 187 controls. The p-value is 9 times 10 to the negative 6, so this isn't really genome-wide significant and not super convincing given the low sample size and not genome-wide significant p-value. Okay, so that doesn't really explain something of interest to me. Another thing that I want to do is to be able to upload a user-specific track. So this is a track that I have about the genome. One track that's really easy to do, to upload, and very useful is just to upload that particular SNP. So to do this, I can create a bed file. The bed file is different than the bed file that Derek talked to you about. Confusingly, in genetics, they use same terminologies for files sometimes. So all I do is I enter the chromosome, the start and end positions of that base pair. So that's my most significant SNP. And then I can label it with a name. So in this case, I'm going to say significant hit. I can then upload this using add custom tracks. So I just click add custom tracks. I choose the file that I was interested, this bed file. And then I can see now I have a new user-supplied track which shows a significant hit. Now I can scroll back and forth on the UCSC genome browser and not expect the SNP to be in the center, but I can know exactly where that SNP is. This is just a useful tool. If you have other bed tracks though, that you're interested in, you can upload those very easily. Okay, so UCSC genome browser is one way of exploring the genetic locus. It's a really nice tool. And it allows you really a lot of power. It doesn't necessarily make publication beautiful pictures. If you want to make publication beautiful pictures, one of the tools that's great to use is called locus zoom. All you have to do to use this tool is to upload your SNPs and your p-values to this website that's given here. Make sure your file, though, is not too big. This is important. If you upload the full genome-wide association stats here, you'll crash their server, / it just won't upload. So all you have to do is upload that path, give the p-value column name, the marker column name, and select the SNP of interest. And when you do this, you'll get an output that you guys have seen multiple times by this point. And these maps are really nice for showing the locus of interest. They're basically a zoomed-in Manhattan plot. On the left axis, on the y-axis, you can see the negative log 10 p-value. On the x-axis, you can see the base pair position, and you can see the gene annotations below that. You can also see the r-squared of the SNP that I selected in the previous screen to all of the other SNPs. So I can see the LD region, the full LD region. These plots are really great for not only publications, but understanding where your SNP is and location in the genome. Okay, so even though I've identified this particular genomic region, I really don't understand the possible functionality of that region. So this SNP that I found associated is not necessarily the causal SNP, but maybe in LD with the causal SNP. So we have found a genetic variant, a SNP associated with a trait shown there, but it may be an LD with known functional variants. For example, a missense SNP, or a SNP in a 5' UTR, or a nonsense SNP, or something like that. So how do I go about finding if this particular SNP is related to any SNPs of known functionality? So again, I can go back to the UCSC genome browser and use another functionality of this called the table browser. So the table browser is selected here at the top. Table browser, instead of just having a visualization, allows you to download large text files from the UCSC genome browser that you can then mess around with in programs afterwards. So I click on the table browser, and then I get a table browser screen at the bottom here. I can select, again, that I want a human genome, that I want the coordinates. Again, that's very important to select the right coordinates. I want to select variations because I'm most interested in those. The track that you want to use is generally common SNPs if you're doing GWAS, but not if you're not doing GWAS, then you don't necessarily need to select the common SNPs. Then you can click filter here to create a filter. What I can do by this is I can select only those SNPs that have known functionality within this table browser. So I can select frame shift sites, either splice sites, nonsense coding synonymous, which doesn't change the amino acid structure of the protein, UTRs, or stop loss, where a stop codon is lost. So I can set that filter so I will get back any SNP that has known functionality. And I can output a spreadsheet of that information. So here you can see an output spreadsheet of this information for the locus of interest. So I can see the chromosome, the start period, the end period, the RS numbers, and whatever the functionality of those particular SNPs are. So now I have what SNPs are functional. Now I need to know, are any variants of known function in LD with the top hit? So how do I get those LD from the top hit? For that, I can use what's called the SNAP proxy search. This is one of the tools that's available to do this. Here, all I have to do is go to this website, enter a particular genetic variance of interest, then set my search options. In this case, I want them to match the population that I'm interested in. So I'm going to match the 1,000 genomes pilot, and then use the CEU, which is the European population, set an R-squared threshold of 0.8, and look for all SNPs which are highly correlated with my SNP of interest. So I can get, now I have proxy SNPs and I have functional SNPs, and I want to say, is there any overlap? Are there any proxy SNPs in the list that are also functional SNPs? Unfortunately for us, no, there are no proxy SNPs that are functional SNPs. So our hit cannot be explained by known functional variants. Okay, so that's disappointing. So what else could it be? So we can start to look at other processes to understand what the potential functionality of the variant is. So one of the ways that you can look at things is to look at epigenetics. Epigenetics are anything on top of the ACTG sequence of the genome. So they can be, for example, DNA methylation, which Sven talked about before, measured through bisulfite sequencing. They can be chromatin modifications where the histones are acetylated or methylated, which open or close the histones and mark areas of enhancers or repressors. They can be potential functional elements marked by DNA's 1 hypersensitivity site. So those are open regions in the chromosome. And within DNA's hypersensitivity sites, which are potential functional regions, they've also measured transcription factor binding sites, so where particular transcription factors are bound. And within that, you can try to see if there are long-range regulatory elements, promoter elements, or if there's transcription at a certain site. On top of that, you can also look for high, large, long-range chromatin interactions. So is this particular part of the chromatin interacting with another part of the chromatin? So if my particular SNP is in one part of the chromatin and interacts with, for example, the transcription start site of another part of the chromatin, I may be able to see that with using these methods. So all of these epigenetic methods are very interesting to do, are all cell type specific, and are often given through the ENCODE consortium. So the ENCODE consortium has studied, I think, over 100 cell types, looking at all of these, or many of these epigenetic markers in all of those different cell types. These are great annotations that are added on top of the ACTG base pairs that you can have. More recently, though, there is now what's called the epigenetics roadmap. So ENCODE consortium just studied cell lines. Epigenetics roadmap has studied many, many more different types of tissue, including, for interest in our group, brain. So they have brain, they have angular gyrus, they have parts of the cortex, they have substantia nigra, and they have hippocampus. They also have fetal brain, which is really interesting for studying development. And they also have ES-derived neural stem cells and ES-derived ganglionic eminence and cortex. So this is another resource that's really going to be interesting to use for studying the epigenetics of the genome. Okay, so you can look at ENCODE within the UCSC genome browser again. So you can add the track here, and you can look at how the different chromatin areas are actually classified. So there's a program that's used to classify the chromatin according to different binding sites or different markers of chromatin. So this particular marker appears to be a CTCF binding site very close to our locus of interest. So that's interesting. It doesn't necessarily demonstrate it as causal, but does say that there could be a regulatory element near our locus of interest. And just to demonstrate that, so here's another way of showing these locus zoom plots. So here you can see basically a standard locus zoom plot, and then zoomed into that locus zoom plot at the high LD region, you can show the different chromatin states. You can see that none of the chromatin states are really classified except for one particular region that's in high LD, which is this CTCF binding site. And here's a website where you can use to make this. So again, doesn't necessarily demonstrate that this is the causal element here, but since it's the only one in the area, it does give us some kind of hints that this may be a causal element within the area. Okay, so since we have potential regulatory elements there, I'm also interested, and I can't really interpret it in terms of known exonic differences. I am interested to seeing if the SNP associated with this trait actually affects the expression of any nearby genes. So do alleles at the risk SNP affect the expression of any nearby gene? And this is one of the ways to try to move from variant to gene. So these are called EQTLs. There are many EQTL databases available, and these are extremely useful for doing interpretations of your variants that you find from genome-wide association. One of the most useful is called Brainiac, which was just released, I think, last year. It is very useful because it has many different brain regions. It has about 150 subjects in it, and it has a really nice web-usable tool for it. There are other different EQTL databases as well. GTEx is a very big whole body EQTL database. However, the brain samples are only about 30 right now. So this is something that will be useful in the future, but maybe not so useful for brain right now. One of the interesting things is that all of the other EQTLs so far really have been done not with RNA sequencing, but instead with arrays. But GTEx is one of the few that's been done with RNA sequencing, so may allow you to study those rare, those lower expression genes a little bit better. Even though it's not brain, the blood EQTL browser is extremely useful as well because there's so much more data. I think there's maybe 10 times the amount of data in the blood EQTL browser as there is in any of these other, any of the brain banks. And there's also an NCBI EQTL browser that hopefully will have a lot more things in the future. Okay, so we, for our study, basically phoned a friend before the Brainiac database was available. We asked collaborators to look at this particular SNP and to see if the alleles at this SNP affected the expression of any nearby gene. And we found that the alleles of the SNP did affect the expression of KTN1 gene. And importantly, we replicated this in 2 different data sets. So this is one data set, and we can see that there's a clear linear relationship between the C allele and the expression of KTN1. And that is highly significant. And then we replicated that in an independent sample in both frontal cortex and in putamen itself. So what we can say is that this SNP, at least in frontal cortex, and we have some hints that it in putamen itself, affects a gene. It's replicated in 2 samples, and now we can try to move from a locus to a gene of interest, right? Again, this does not necessarily demonstrate that the causal reason behind putamen volume is through this. However, it does give us some pretty strong hints about that. Okay, so now that we have a gene that we're interested in, we want to use other tools to see where and when that gene is expressed in the brain specifically. So, okay. So one thing to keep in mind is that 86 to 95% of genes are expressed in the brain at some point during the lifespan. And 90% of those are differentially regulated in region, across region or time. So, very importantly, expression of a gene in brain does little to implicate it as causal. So, don't necessarily pick the gene that is expressed in brain because most genes are expressed in brain. Don't necessarily pick the gene that's close to the locus of interest because it is differential during some period of brain development because, again, most genes are differential during some period of brain development. So, that's important. However, it is interesting once you have identified a gene of interest to look at that gene expression across time. And it may lead us to cell type hypotheses. For example, if it is only expressed in a very early period of corticogenesis, it may be involved in progenitors and not in the neurons. So it may give us that the progenitors are particularly the cell type of interest for us. So to do this, you can use a tool called BrainSpan. BrainSpan can be found here. It's provided by the Allen Institute. You heard about it a little bit before. You can click on this developmental transcriptome, type in your gene of interest, and you can see the expression either by time or by region. So you can see here that they're organized by the time. So they go all the way from 4 post-conception weeks, so prenatal, all the way to around 80 years old. So this is a really great resource to see the expression across time. This is the actual expression of that gene. You can see that it doesn't really change too much by time. It doesn't seem like it has really high at one end or really low at the other. So this doesn't really say that time is like the predominant driving factor for the expression of this gene, nor does region. So you can, they've sampled regions from multiple regions of the cortex, multiple subcortical regions, and cerebellum. And again, it doesn't seem like it's expressed in any particular region more than another. Okay, so that's basically exploring a locus of interest. Now I'm going to talk to you a little bit about gene-based tests, which are ways of collapsing, combining SNP associations across genes to form a gene-based p-value. So when you do a genome-wide association study, you get you get SNPs in all regions, including intergenic regions, and you get their negative log 10 p-values. And you can see that some genes may have more association than others. So there are advantages to doing these type of tests. One of the advantages is that you have greater interpretability, right? So these gene-based tests allow you to look at the functional element of the human genome, the actual gene. They have fewer multiple comparisons because there's only around 20,000 protein coding genes in the genome, whereas there's millions of genetic variants that you can study independently. And they can easily feed into pathway-based approaches that I'll talk about in a second. However, there's some very serious disadvantages that you should be aware of when doing a gene-based test, which is that first it ignores intergenic variation, so most of the genome is intergenic, so you're just throwing that out. It generally ignores the direction of association, depending on the type of test you're doing. So, for example, some tests can just look at the p-value. You can have a very strongly significant positive influence on the trait on this end of the gene, very strongly significant negative influence on the test on this end of the gene, and those are just, the directionality is not taken into account at all. It ignores that a variant within the intron of one gene may be affecting a totally different gene. So you can have a significant SNP here, which is in an intron of the gene, that totally affects the expression of this gene. But the model that you're using does not say anything about that. So it's an important disadvantage to realize. And there's actually some really interesting examples of that. I think FTO is one of the examples. Okay, so there are many tools for gene-based tests. Some of the tools, all the tools use different statistical ways of combining SNP p-values to get a combined gene-based p-value. One of the tools corrects all SNP-based p-values within a gene for the total number of independent tests, then takes the minimum p-value across that gene. That's implemented in this Gates algorithm, and you can do that through KGG. And there's a tool to do that for you, so you can just go there to get gene-based p-values, and of course accepting the caveats. Okay. Clicky. There we go. Okay, so a next thing you can do is do pathway analyses. So if I know that there are particular genes involved in a biological process. So for example, mitosis is a very well-studied biological process. There are known genes involved in mitosis. Some of those are included here. So I have a list of genes, known genes involved in a particular process. And then I also have genes that are associated beyond a certain level with my trait of interest. If I overlap those two lists and I find that there's an overlap significantly different than I would expect by chance, then I can say that it's likely that genes, that this process is involved with whatever phenotype I'm interested in. Okay, so this is a really great, has amazing interpretability, right? So we can come up with novel hypotheses about what the genetic influences on a particular trait are, what the biological pathway is for that particular trait. It has amazing interpretability, however it has disadvantages, it has all the disadvantages of the gene-based tests, and it's also that most of these gene lists are not really well known. Okay, so here's two tools for pathway analysis. They don't use the exact Fisher exact test that I showed you before, but it's generally a similar type of analysis. So those are two tools that you can use if you're interested in pathway analysis. Okay, so those are just some ways of understanding and going beyond doing the genetic association to looking at what the locus is and the functionality of that locus. So conclusions: identifying the genetic locus is a causal foothold into understanding novel biological mechanisms. There are many databases and tools that will allow you to form hypotheses about biological mechanisms, so really feel free to explore them all. However, it's easy to make a story. Let the evidence guide you. It's very important to let the evidence guide you. So I'd like to thank Derek and Sarah, and especially like to thank JB and Tom for helping me organize this session. And thank you guys for listening.
One quick question before we end the session. No? Yes? Well, if there's no question, I mean, let me thank again all the speakers. Let me thank Jason, who was really the driving force with this course. And please do give us feedback. We need to see whether, how we go further with this course and what are the different things we should be doing better or we should not be doing. So please give us your feedback on the course, and you will get the slides on the OHBM website. Tom will put them as well on his website.
Right, yeah. I just wanted to repeat that the slides will be available, I think, 3 ways. So one is the OHBM website. I know there's only 2 on there now, but we'll put the rest on there. The second is that they're filming all of this, so that should be available. And the third is Tom Nichols' website has all the OHBM imaging genetics course from years past, and we'll have this year's too.
And thank you for coming, and let's hope that next year will be even better.